Good morning, everyone. Welcome to the Minor Use Foundation Global Residue Studies Field Research 101 webinar. And this morning, Joe DiFrancesco, Wilfredo Robles, and myself, Stephen Flanagan, will be presenting some basic background information on conducting residue trials. This will be the first of 6 webinars, and we will have breaks this morning in between the presenters so that we can take questions, and you can also add questions in the chat and raise your hand if you need to in case we didn't get to your question. Okay, so next slide, please, Nick. So today we're going to talk about the basis of conducting field research trials under GLP OECD guidelines, the protocol, and the notebook. Next slide. And Joe is going to cover GLPs. Wilfredo is going to cover protocols. I'll talk in broad terms about the notebook. As I mentioned, we'll have breaks in between for questions, and we'll start with a little bit of an introduction. So next slide. So let's say you want to bake a cake, and you want your cake to be a fairly good cake. You want it to taste nice for your guests. What do we need to bake a cake? Slide. Okay, what do I need? What do I need? Next slide. Well, I'm going to need a recipe. I'm going to need to know, an idea of what I'm going to do. I'm going to need some ingredients. I'm going to need a kitchen in order to prepare things. I'm probably going to need an oven. And there's techniques that are involved in successfully making a cake. Next slide. It's kind of similar if we're going to, make a field trial, right? We have a recipe, which is a protocol. We have ingredients. You've got a field, you've got a crop, you've got the applications you're going to make. And then we have a kitchen, and the kitchen could kind of be considered, it's the field where all this preparation happens. It's the sampling, and then it goes into a freezer. And maybe the oven is the actual lab where we cook the samples and we look at them. And then the techniques really are the GLPs, the SOPs, the operating procedures, and really the techniques are your experience as a field researcher, knowing that crop, knowing how to grow that crop, knowing how to successfully conduct a research trial that's going to get the cake that you want. So next slide. And that process has a bunch of different pieces. It has, if you think about it, it might be second nature if you've made this cake 100 times, but if it's the first time that you've made this cake, it's going to take some careful planning and getting all these pieces together so that what you end up with is this nice cake. Next slide. And essentially, if we wanted to just get a piece of carrot cake, we could go down to the bakery. But in our case, what we're going to do is we're going to produce these 2 crops. One, they're going to look exactly the same, but one of them has this invisible ingredient, namely the new compound that we're trying to register. So essentially what we were going to do is we're going to produce 2 different cakes, one with the actual compound and one without, and then we're going to compare them, and that's going to be the data set that we're going to develop. So that's just a brief introduction, and now we're going to hand off for our next section. Next slide. Oh, yeah. So what does a residue trial deliver? It's the notebook, it's the samples, and then it's the support for the use. And the next slide will be for Joe.
Okay, thanks, Stephen. Yeah, I'm Joe DiFrancesco. I'm retired from Oregon State University in the United States and have been a part-time consultant with the Minor Use Foundation for several years now. And so this morning I'm going to talk about GLPs. Next slide, Nick. Next. GLPs stand for Good Laboratory Practices. What are they exactly, and why are they so important? Next. Basically, GLPs are a set of guidelines. These guidelines cover the organizational process of a study, and all the conditions under which a study is planned, performed, monitored, recorded, and reported. So basically the entire study from the planning stage all the way until the samples are collected, the data is analyzed, and the reports are written. Next. GLPs promote good science and good data. So when you evaluate a chemical, in our case, pesticide, the data has to be of high quality and reproducibility. Otherwise, the data set is not valid. GLPs, you'll see, help promote the development of quality test data, and that's what we're after, quality test data. Next. So don't be confused by the name. They're called good laboratory practices, but they do apply to the field operations in a residue study. The idea is to promote quality and validity of that test data, especially for a pesticide that's going to be used on a food commodity. So in general, they help ensure uniformity, consistency, reliability of that data, reproducibility of the entire study, quality and integrity of that data, which are all imperative when you're conducting a residue study. Next, please. So a little bit of history. The GLPs that we use at the Minor Use Foundation for Residue Studies were developed by the OECD, the Organization for Economic Cooperation and Development. They got together in 1961. 38 countries got together to create this organization, and their goal was to promote and stimulate economic progress and world trade. And one of the ways they felt they could do that is by developing these guidelines for studies that are conducted internationally to ensure that the data is of the highest quality. So it's a set of standards, and they are accepted internationally. Any study that the Minor Use Foundation conducts adheres to GLP principles and guidelines, as do most international residue studies, whether they're conducted by us at the foundation or the registrant or the country itself. They will adhere to GLP guidelines. There's a screenshot on the left. That's the very first guideline that the OECD developed back in 1995, revised in 97. And there's a URL there that you can go to review this document. And I think Nick might put that URL up in the chat. He did, yeah. So if you want to refer to that, you can go there. Thanks, Nick. Next slide, please. So what are in these guidelines? The first guideline they developed on the principles of good laboratory practices. Well, the general topics that you'll find in there, first one deals with organization and personnel. Organization of the study itself, the people involved in the study, clearly delineating their roles and responsibilities, and most importantly, making sure that the people involved in the study are trained to conduct and to participate and do the activities in that study that they're responsible for. An important component of GLPs is having a quality assurance unit overseeing the study. This is a person, an independent observer, who monitors the study for adherence to GLPs. Every study conducted by the Minor Use Foundation will have a quality assurance person assigned to that study, and that person is responsible for observing different aspects of the study as it's being conducted. For the field, that person will go out with you and observe an application. They might go observe a harvest of the samples to make sure that they're done properly according to the protocol and that you're using good GLP guidelines while you're doing the activity. So that person is independent and is an observer in the study. Facilities is another topic you'll find in the guidelines. This is for the field and the lab. Making sure that it's adequate in size, that it has all the equipment that's needed to conduct the study. Is there a spot to store the data someplace secure and safe? That's an important part of GLPs, is making sure that that data is safe and secure. Facilities also apply to the field in a sense. Are the plots set up to minimize contamination from treated plot to the untreated plot? For example, making sure that the untreated plot is upwind from the treated plot, so there's no possibility of drift when you make the application. Equipment and instruments, any devices used in that study need to be inspected, maintained properly, of course, calibrated before they're used. The reagents used in the lab, the pesticide used in the application needs to be labeled properly and stored properly too. The test substance that's used in the field, for example, needs to be monitored for temperature during its storage from the time you receive it at your facility until the time of the last application. At the end of the study, you will have a printout of the temperatures, or you can record it with the min and max thermometer. We want to make sure that the test substance has not lost its integrity during storage, making sure it didn't freeze or get too hot. So in storage, and also when you're taking that pesticide out to the field to make an application, temperature needs to be monitored to make sure that the test substance maintains its integrity. If somehow it loses its integrity or its potency, of course, that will affect the residues at the very end. So maintaining the integrity of the test substance is very important. Standard operating procedures are a requirement in GLPs. These are procedures that are standard for your facility and for that study. Things like, how do you calibrate your sprayer? How do you collect samples to ensure that there's no contamination? These are common procedures or standard procedures. And every facility will have a set of these procedures that they follow. New people involved in the study could read these procedures to know how things are done at your facility. And the quality assurance person will look at these SOPs to see if you're adhering to your procedures that you said you would do. So every field office will have a set of standard operating procedures. And lastly, the protocol or study plan itself, making sure that contents in the protocol are clearly stated, the objectives are clearly stated, and the methods to obtain the objectives of the protocol are clearly outlined in the protocol, so everyone knows what they're doing and they can follow the methods that are outlined in the protocol. So these are some of the general topics you'll find in the GLP guidelines and things that the quality assurance person will be looking for and things to keep in mind as you conduct your trial. Next slide, please. The OECD has been busy over the years, starting in the mid-90s up until recently. They've developed 25 different guidelines, part of a series dealing with GLPs and compliance monitoring. The last one, number 25, was just issued December in 2024, and it's a position paper on good laboratory practices and IT security. So the OECD is trying to maintain relevance and current with the times in dealing with IT security. There's also a link here, a URL that will take you to all 25 of the guidelines in the series if you were interested in looking at the other topics. Next slide, please. Data integrity is of utmost importance in a residue study, and the OECD realizes that. They developed a guideline number 22, dealing with data integrity, and they want people to adhere to the ALCOA principle, and that's an acronym. The A stands for attributable. All the data in a study needs to be attributed to a specific person. Anybody generating that data, modifying the data, reviewing that data, that person needs to be identified in the field data notebook. L stands for legible. That's kind of obvious. The data entries need to be legible so anyone can read it. C stands for contemporaneous, meaning that all data needs to be recorded in real time, not at your convenience or later or when you get back to the office, but when you are collecting data, you need to record it in the field data notebook in real time at the time of occurrence. O stands for original or original records. Original records get included in the field data notebook, or if it's a copy of a document, it needs to be verified that it's a true copy of the original. And A stands for accurate, which again is obvious. We want to make sure all the data is accurate. So keep Alcoa in mind when you're conducting these studies. Next, please. Documentation and verification are very important in GLPs, starting with the study plan. It's reviewed and signed by the sponsor, which is the Minor Use Foundation, the study director, the person who wrote the protocol, and the quality assurance person. So all three people review and sign, indicating that they all know what's in the protocol, and the protocol is easy to follow and will fulfill the mission of that protocol and get the results we're looking for. GLPs, of course, deal with any equipment or instruments used in a study, and documentation is important. You need to inspect your equipment, clean it, maintain it, calibrate it. But there needs to be a record that you've done these activities. We have a saying in GLPs that if it isn't written down, it didn't happen, meaning that you do these things, and you probably do that in any field trial, but it needs to be documented. And that's a very important component of GLPs, documenting everything that goes on in a study. Test substance needs to be labeled properly, of course, and as I mentioned, temperature monitored. And the contemporaneous idea is very important, that entering data at the time of occurrence is imperative. It needs every bit of data that's entered in a field data notebook needs to be signed or initialed and dated by the person collecting that data. Next, please. So these are what GLPs are, and this is why they're important. Any national regulatory authority that's going to register a pesticide will require that the data that they're reviewing is conducted under GLP guidelines. One country might accept data from another country that was generated in another country, but only if the data was collected using GLP guidelines. That way they know that the data is of the highest quality and has integrity. If the data is good, pesticide is registered, safe to use, that of course helps remove any trade barriers that might exist, and also protect human health and the environment. And lastly, the Minor Use Foundation adheres to GLP guidelines when they conduct studies, and the Codex Committee on Pesticide Registration will not entertain or review data that has not been conducted under GLP, and that there's evidence that the study was conducted using GLP guidelines before they will establish an international codex somewhere else. So they're very important for a whole host of reasons. But not to fear, Minor Use Foundation staff is on hand to any questions you might have about GLPs during your study or now. I think we're taking questions. If you have any questions, please feel free to ask them or send us an email or wherever you, but during your study, we will be available to provide answers too. Thank you.
Thanks, Joe. And one of the things I've neglected to mention is that I'm also retired from the University of California, Davis, where I worked with the IR4 program and did a lot of studies and work with Joe and Wilfredo in their different regions. And I think one of the real guidelines here in terms of many of us come from field research backgrounds where we do efficacy studies and we are very familiar with replicated designs and the details of a replicated study. And one of the things with GLP studies is that the documentation is really the bigger part of the work in many regards in a residue study than it is in an efficacy study. So there's a little bit of a translation that has to happen when you go from being a efficacy researcher into this world of GLPs and residue studies. So that's just a little bit of a background. Any questions from Joe's slides?
You see Jeffrey has his hand up.
You can go ahead and unmute yourself and ask the question or type it in the chat, either way.
Yeah, thank you, Joe, and thank you, Stephen. Joe, thank you for your presentation. Mine was just a follow-up question on the temperature monitoring of the test substance. At the point when you have already mixed the test substance and you're waiting for application, I know there's always some bit of time you take before you start the actual application. Are you supposed to also take that temperature? And then what is the appropriate time you are supposed to wait before you start the application, the maximum time? Thank you, Joe.
Yeah, thanks, Jeffrey. So the test substance container that contains the undiluted pesticide needs to be monitored during the whole length of storage and while you take it out to the field. Once you mix it, we don't need the temperature of the mixture, but you do need to apply that mixture within 2 hours of mixing. Did that answer your question?
Yeah, thank you for that. Yeah, I think that is clear now. Yeah.
Okay, you're welcome.
Okay, any other questions out there? Onesimus, you'll need to unmute yourself.
Good afternoon. In Kenya, we are in the evening. And good morning. My name's Onesmus from Kenya. My question is the use of adjuvants during the application of the test material. Because I think most of the protocols speaks about adjuvant where you use it, you have to record. Maybe you can speak a bit about the adjuvants. Thank you.
Yeah, thanks, Onesimus. I'll have to get a confirmation from Stephen and Wilfredo. If I recall, most of our protocols will say you can use an adjuvant if it's a common local practice. Some protocols require an adjuvant, and it will be listed in the protocol. They want you to use an adjuvant, and they'll tell you whether it's a penetrant or a sticker spreader. I think most of our protocols, though, say that you can add an adjuvant if it's a common practice. I don't know, Wilfredo and Stephen, am I correct on that? I'm trying to think of a recent protocol.
Well, it has to be indicated by the protocol. That's, I mean, so some of the protocols required the use of adjuvants, and some of them not. So it all depends.
Yeah, I know that if the registrant wants an adjuvant, we will list it in the protocol. What I'm getting at is, it seems like we have a statement in there that says you can use an adjuvant if it's a common practice in that particular crop. But I'm sorry, I'm not positive about that. Onesimus, as with any study, if there's ever a question like that that's not clear to you, you can contact one of us or the study director directly, and that the study director will answer that question.
Yeah, I was asking because there's a time we are asking ourselves because of the weather at a given time. Can we use adjuvants or not? Because it was there, as you are saying, in the protocol that you can use. So it was
Oh, yeah.
a choice. So, and the weather was not good at that time. So we were asking ourselves, can we use it? But good enough, it did not disturb us. So thank you.
Yeah, the protocol will say use it if you want to. But like I said, if there's ever a question and you're in doubt about something, don't hesitate to shoot a question to anyone within the Minor Use Foundation.
Okay,
Thank
and
you.
since we're talking about protocols, that's a good segue to Wilfredo, who now will go ahead and talk about the protocol.
Yeah, that's a good way to start my presentation. Next, please. Because to me, I mean, the next slide, Nick. To me, the protocol, I'm a big Star Wars fan, so to me, the protocol is the Force. For those of you who are really fans of Star Wars, you know that the Force is which this mystical energy of field created by all living things. So I mean, that's just a metaphor related to the protocol. I mean, what the protocol says is what you're going to do. I mean, some protocol says don't use surfactants. Some of them will say use surfactant as recommended by label, and some of them will say use surfactant and a certain percentage of volume by volume mix, total mix. So it all depends on the protocol. So everything has to be rely on protocol because that's the truth. Go ahead. Next, please. So in general, the protocol is a written document where the objectives and experimental design are described to conduct the study. I mean, it's composed by 35 sections, and prior project initiation, you need to ensure once you get that protocol, and that is already signed by the sponsor representative, in this case, Minor Use Foundation representative, and also by the study director. I mean, the study director is a person who oversees the entire protocol and responsible for the overall conduct of the study. So in this case, I mean, if both signatures are not included on that protocol, I mean, there's no way to conduct that trial in the field. So that's the official way to start the protocol. Next, please. Once you get the protocol, there's some nomenclature that typically is being used in the protocols. I mean, first, I mean, on the right top corner, you will see a study number. In this case, I put as an example, 000, and date of issue of that protocol, it's December 2024. So in this case, you're gonna have different numbers and different letters on that protocol to refer that the signed protocol. So on the first one, the first three numbers is related to the study number. On this case, 000. Then it's the 24, which means it's a study year. So that way, I mean, everyone related to this protocol will know, I mean, when the protocol was signed and when it's expected to be conducted. And then the third layer or third acronym is the country. I mean, who is conducting this trial? On this case, I use United States as a two-letter acronym or symbols, but it can be Puerto Rico, which is PR. So, and 01 is the trial number, which belongs to the same study number. So you can be conducting a trial under a study number 000, but the assigned trial number to you is 0203. It all depends of how the study director, I mean, display this kind of nomenclature. Next, please. So once you get that protocol, in general, the first 6 sections will be related to how the justification of the protocol, proposed dates, and also a study director. I mean, you will see on that sections 4, I mean, who is in charge of this protocol. I mean, this is pretty important for you because this is a person that oversees the entire field study. So if you have any questions related to the protocol, just ask the study director always. I mean, before making a decision in the field. Then section 7 through 9 is more related to quality compliance and quality assurance and GLP. So it's basically a general description of how it's going to be conducted. Next. To me, as a field person, I forgot to tell you that I'm a field research director for IO4 project in Puerto Rico. I'm professor at the University of Puerto Rico. So I've been conducting this kind of GLP trials since 2011. So to me, from my experience, I think these 3 groups of sections are the most important section to be able to conduct the trial in the field. So the 10, 11, and 12 is more related to the test site. This is where you're gonna have a description of how the layout of the field plot and how those plots should be arranged in the field. Test system design and preparation. And then the test substance. I mean, how you're gonna handle the test substance, how you're gonna be applying those treatment, how you're gonna, what rate you're gonna be using for the test substance. So those are 13 to 16. They always give you some supplemental crop treatments, for example, surfactant. I mean, if surfactant is needed, this is the session that you're gonna be seeing this kind of information. And then samples. I mean, how you're gonna collect the samples, when you're gonna be collecting the samples, and how you're gonna be handling the samples and shipment. To me, this is pretty important because despite you're conducting an excellent work on the field, applying those treatment, I mean, the way that should be expected on the protocol, if you don't collect the samples, appropriate samples, I mean, on the field, and you basically don't meet protocol requirements, basically the entire study will fail. So ensure that the samples are harvested and processed the way that is written on the protocol, despite, I mean, how successful you were applying those treatment on the field. Next. Section 20 is more related to field documentation and record keeping. I mean, basically, it's driven by trust. So as Joe was explaining, you need to record the data right away, promptly, and directly into the field data book. I mean, there's no separate piece of paper that you can have that information logged. So had to be sufficiently detailed. And if you basically make a mistake, which as a human being we can make, I mean, just say it. I mean, say that the date that we record the application date was wrong, and you basically have those codes that you can use, like on this case. I mean, we wrongly put the wrong date on this case as anticipated last application date as 10/17/2017, and then when we recognize the error, we basically cross it out with line, put a error entry code, and then initials and date, and then you correct. I mean, you cannot erase those numbers. Just tell, I mean, how you make the mistake and how you correct the mistake. Next, please. So section 21 is more pertaining to amendments and deviation. An amendment by definition is any intentional change from the protocol after study initiation date. So for example, when the study director signed the protocol, by then they didn't know, I mean, which laboratory will be processing the sample. So they can assign the protocol and then assign the lab that's going to be processing the sample. I mean, they issue basically an amendment to the protocol. You can conduct the trial in the field, start the trial, and then the study director will make this amendment and assign the protocol later. So that's completely fine. And deviation, this is more pertaining to field people. I mean, any person can use this choice in case of you have any unintentional change from the protocol. For instance, I mean, equipment failure. Things happen in the field. The equipment fails, malfunction for sample processing, and you need to fix that. So this is out of your hands. So basically, you explain what happened, send it to the study director, and then the study director will make the decision how to proceed afterwards. And also plant disease outbreak. This is just an example. Go next, please. For instance, on this left photo, this is plant disease, coffee berry disease. A couple of years ago, we had an outbreak, and we had a low sample collection. So we issue a deviation because we didn't meet the sample size because an outbreak. So I mean, the study director made the decision and basically clarified that the sample D or sample C had a lower number of pounds of this coffee berry. And then on the right side, probably some of you have known, I mean, in 2017, Puerto Rico was beaten by a catastrophic weather event, which is the Hurricane Maria. I remember pretty well. I mean, we had like 5 trials in the field. We got hammered by this hurricane. So some of the field trials had to well, take it out from the field, and basically they take it out, and we had to redo it the next year. But some of them were saved and were able to send the samples out. So I mean, it all depends. This is basically issuing a deviation that due a catastrophic event, I mean, we couldn't finish or complete the trial. Next. So section 22 and 23 are more related to field personnel and archiving. I mean, how the originals will be turned into minor use foundation. Next, please. And 24 through 30 is more related to the lab. I mean, this is where the protocol describe how the samples should be processed and should be analyzed by the lab personnel. So basically describe the methodology, analytical, and how they're going to be conducting those samples. Next, please. This one is basically similar to section 21 for the field people. This is for the lab. Basically, is recording, I mean, the issue of amendments or any deviation that perhaps the lab can conduct. Next, please. And then the data record for the lab personnel, laboratory documentation, record keeping, basically archiving is basically described on those 3 sections. Next, please. And then the last one is basically administrative indications that how the protocol amendments and deviation, final reports, any raw data will be retained, has been retained by the study director and shall be archived by Minor Use Foundation. So it's basically a process. So just lead through and remember that the protocol is a force. It's the force that basically move everything on the field and the lab and the administrative personnel at Managers Foundation. Thank you.
Thanks, Wilfredo. And we'll pause now for any questions about the protocol. One of the things I was thinking when you were talking, Wilfredo, is that the protocol goes through a lot of iterations or changes and refinements before it gets out into the field. But just like any human process, there are errors, there are things that may not be clear for your exact field site. So that's where communication with the study director and the team is super important in terms of clarifying exactly what is required. So always communicate and ask questions. It is part of the process and very important.
There's Joffrey, raise the hand. Joffrey?
Yeah, thank you very much, Wilfredo. Just two questions to follow up on your presentation. One is with regard to the amendments. When it comes to deviation on the sample size itself, given the weather or changes, I just wanted to bring your attention to a codex document guiding on the sample size before you really sample. How do you balance between the codex guidance document on the sample size and what is in the protocol itself, given the circumstance that might arise during, say, the field when there's a weather change affecting the residue level? Then the other question to you, which I also wanted to raise, is on the plot plan. Is there a specific plot plan which the protocol demands really a field study director should follow really in terms of the plot plan itself? Because what I know, there is that element of the plot plan, but there's also the direction to the plot and all that coming up in the initial write-up of the protocol itself. So maybe you need to guide further on this. Thank you.
Yes, let me see if I can follow those two questions. I mean, I know that those protocols are issued, I mean, based on Codex guidelines. So basically, the study director and Managers Foundation try to meet, I mean, all what is required by like sample size and the way that it should be applied in the field, the test substance. But in terms of the samples, I mean, typically for field layout, for example, what I do is I look for the minimum trees, for example. The protocol will indicate you, hey, you need to, in order to harvest or collect the sample from a minimum of 4 trees, for example. I'm using avocados as an example. So I use that as a starting point to lay out my plot. Instead of using the minimum, I try to use 2 or 4 more, like for example, 8 or 6 or 8. That way I can have room space for in case of outbreak or something happen. I mean, and that's how I lay out my treatments in the field. I mean, I try to meet the protocol requirements, but go a little bit more than is required by the protocol. Is that clear, or you need?
Yeah, I think that's now clear.
Yeah.
Yes, yeah, because it helps with the balancing with the codex document also. Yeah, thank you.
Yeah.
And then did you want to address the plot plan question, Wilfredo?
Yeah, the plot plan, yeah.
Will you send the question again? I don't recall it. Sorry.
Yeah, the question was, you know, in the protocol at times there is emphasis on direction to the test plot area.
Mm-hmm.
To the extent where you need to get the GPS and all those details. and then you need to align it to the actual plot plan. So I don't know, maybe because at times one thing I've noticed to the GLP studies, when you overlook one element, then it comes back to really an audit issue, really.
Yeah, you to meet, for example, it's required that you have a buffer zone, for example. It's required that, as Joe stated, that you have your layout upwind. I mean, basically the untreated on the north versus the south, which is the prevailing wind coming from the north. And how that's how a layout, I mean, try to follow that, because otherwise we'll contaminate one plot with the other. So yeah, buffer zone, I mean, the way that is set up on the field, I mean, it all depends. I mean, if you want to send me an email, I can help you out with that. I mean, be glad to help. I'm gonna put my email on the chat just in case someone have questions in the field.
Yeah, thank you.
Yeah, thank you. No problem.
Okay, thanks, Wilfredo, and thanks for the questions. And one thing to point out is that we're having a discussion today at a fairly high level, and then we'll, as we go through in other webinars and other trainings, we'll get down to more specifics. And often the challenges are in the details. I'm going to talk about notebooks now, and I'm just going to give a few kind of notebook page examples. But clearly, a notebook can be hundreds of pages in its completion, and so today we're going to have a discussion that's quite general, and then as we go forward, we'll have more specific discussions about documentation, et cetera. So, okay, next slide, please. So the notebook has different sections or different parts. These are the 9 different parts of the notebooks, basically following the progress from the GLP standards all the way through what happens in the trial and what were the weather conditions that occurred with that trial. So let's take it part by part. Next slide. Essentially, the GLP portion of the notebook has the GLP statement, which is essentially the field investigator stating that, yes, I have been the investigator in this study and I've conducted it under GLP. So this goes back to what Joe was talking about in terms of reproducibility, and essentially you're putting your reputation and your professional qualifications on the line, saying this is being conducted under GLP. In this section 2, you'll have a copy of the protocol and then any amendments or changes, so that, for example, if there's a QA inspection of your site and they have a specific question about this study, you can go right to that section in the notebook and refer to, this is the protocol I received, this is what I agreed to, and these are the amendments and how I applied those amendments. So it's all the details in terms of the conduct of the study and the protocols that drive the whole study. Next slide, please. The second section is the personnel. Who was the field research investigator, and then who else worked on the study? In some situations, all of the documentation in the notebook will be done by the field investigator, but in other sites, you might have technicians who are highly skilled, highly trained, might help in the data recording. We want to be able to see who did what and who actually might have signed into the notebook. So this is again, it's thorough, it's reproducible, it's contemporaneous, it's all the details of who actually conducted this work. Next slide. Communication. This section of the notebook usually has 2 different types of entries. There's obviously kind of a handwritten chronicle of who you talked to, what questions arose from the protocol or the study conduct, and how they were addressed in communication. But then also behind this handwritten log, there's going to be copies of emails or any other communications that were a part of resolving any questions. And again, this is a record so that someone can go back and look and say, oh, okay, so there was a question about an adjuvant use and it was approved. Or this is an insecticide study. They had a disease problem, and this was where they got the okay to treat the disease problem in the field. So this is, again, a whole piece of completeness of putting the whole data set together. Next slide. The fourth part of the notebook is the test substance. And in this portion of the notebook, you're going to see a certificate of analysis. You're going to have a document that tells you what is the test substance that you received from the manufacturer. And there's something of a verification step in there. You want to make sure that what your documentation says matches the protocol, matches your understanding of what you're applying and what that compound is. One of the details here is that oftentimes certificates of analysis have a code name for the product that's not necessarily the chemical name that's in the protocol. So if those names aren't clear, then that's a really important piece to verify before you make any applications in the field. In addition, this part of the notebook is going to have your storage conditions that we talked about and a use log documenting when you took material out of storage and used it out in the field. And that should match up with the application sections in the notebook as well. All right, the next slide, please. Section 5 is the trial site. There was a question about mapping. This is obviously a hand-drawn map. Again, this part of the notebook can have a combination of things which are hand-entered descriptions, and also it usually has some sort of a Google map or a digital tool that shows where the site is as well. GPS can be used in this. You can use Google Maps to show how to get to a site. You can also blow up Google Maps and actually show the actual exact location. As well, you're going to have information in this section about the soils of the site, the maintenance, and the history behind that plot. So again, it's a completeness of data and for the trial site. And the next slide, please. Section 6 is the application section. Much like Wilfredo talked about the sections of the protocol that are so critical to the field components of the trial, I always think of application 6 or section 6 as being this critical piece of the conduct, right? Because if we didn't mix the compound correctly, if we didn't apply it correctly, and if that doesn't match up with the protocol, then it's all for naught, right? So this whole section is really a detailed story about what you did in the field, how the application went, and whether or not it matched the protocol. So this section in particular is going to have a lot of eyes looking at it, making sure that what we stated in the protocol actually happened out in the field. And it's also from a field researcher's perspective, it's also really satisfying when we execute this correctly, right? It's the compilation of everything you've done to prepare and be ready and have things in line to do the application correctly. So it's a critical piece, and it's documented very precisely in this section of the notebook. And again, this is something we'll go into more detail at the other webinars. So next slide, please. Section 7 is sampling, and basically, when was it sampled? How was it sampled? What portion of the crop was sampled? How was it stored? And then how was it prepared for shipping? So again, detail, detail, detail. Next slide. And then section 8 is the shipping piece of it. So making sure that there was a documentation of who took possession of the sample, that it was frozen, and how it got to the lab, whether that was a direct delivery or if it was shipped internationally or however it was to get it to the laboratory. And the next slide. The 9th section is essentially the data supporting that you had a fairly normal or even unusual events in terms of the weather and the growing conditions for that given year. So you'll have a combination of answers, hand prompts that you'll answer, but also you're going to have data in here that is more than likely downloaded from some sort of a meteorological data source. Okay, next slide. Thanks for attending the first minor use webinar on Residue Trials 101. We will be having a follow-up webinar in February, and at that URL you can see the date when we announce it. The next session will be looking much more specifically at test substance receipt and data recording, and as well, this webinar is being recorded, and so it will be available in case you want to reference it at a future date. So, with that, any quick questions on the notebook? Okay, doesn't look like it. So thanks for attending. And
Stephen, there is a hand raised.
Okay.
Hi.
Yes, go ahead.
Hi. My name's Kalinka. I was a little late for the webinar actually, but I just wanted to ask with regards to the communications log, you mentioned that should there be any loss of samples due to pathogens, would you refer in handwriting to the deviation?
Sure. There could definitely be a note in the communications log about how you communicated the problem, and then the actual deviation would be in this GLP section in terms of once things are resolved and you've submitted a deviation.
All right. So does the study director also sign off on the communications log then?
Not necessarily. I mean, there'll be pieces from the study director in terms of communication back to you. So you ask a question of the study director, they answer back. That can be in the email trail, if you will, in the communications log, but they don't necessarily sign it.
All right. And then just one last question. You also mentioned the storage conditions. So I saw with regards to the temperature, are you supposed to show that what the actual temperature was with like data loggers or something like that?
Correct. Yep.
Okay.
And there's various different ways to do that. It can be just as simple as a max min, excuse me, or more detailed with a data logger sent to record every, hour or whatever.
Yeah. All right. Okay. Thank you so much.
Okay, and we'll cover more of those details as we go forward in the webinars, but thanks for the question, and thanks everyone for attending today.
Thank you very much.